Hello everyone, good afternoon and welcome back from the coffee break. I am going to start session 3. Hopefully we can take our chairs and can start our session. So hopefully you have enjoyed the presentation and panel discussion from session 1 and 2. And we are now entering the last session of the day, that is session 3, and I will be moderating this session. I am Khondoker Alam. I work as a senior staff fellow in the Division of Quantitative Methods and Modeling within Office of Research and Standards in OGD FDA. We have 4 speakers for this session, and our speakers will be talking about the potential impact of reformulation on the bioequivalence. In this area, we have a couple of FDA research going on, and our presenter will talk about and present their research outcome today. And we'll also discuss about how to utilize modeling and simulation to evaluate the bioinequivalence risk in the event of reformulation. And at the end, we'll also talk about the regulatory perspective on controlling NDSRI and how to control the potential bioequivalence approach for the reformulated generics. So I want to introduce our speakers briefly. Our first speaker is Dr. Chris Bode. Dr. Bode is serving as Vice President of Scientific Affairs in Pharmaron, which was previously known as Absorption System. Dr. Bode has directed many in vitro studies in support of BCS-based bioevers. Today, Dr. Bode will present some of the study results which were generated as a part of FDA research contract. Our second speaker is Dr. Sook E. Dr. E is an assistant adjunct professor at the Department of Bioengineering and Therapeutic Sciences at University of California, San Francisco. Her research project focuses on genetic determinants of anti-diabetic responses and the role of membrane transporters in drug disposition. Our third speaker for this session is Dr. Fang Wu. Dr. Wu is a senior pharmacology reviewer and scientific lead for oral PBK modeling in the Division of Quantitative Methods and Modeling within Office of Research and Standards. Dr. Wu is responsible for using modeling and simulation tools for reviewing pre-ANDA meeting packages, ANDA consults, and control correspondences. Our last speaker for this session is Dr. Dongmei Lu. Dr. Lu is serving as policy lead at the Office of Policy for Pharmaceutical Quality within OPQ FDA. Dr. Lu has extensive formulation-related experience from working different pharmaceutical companies. Before joining to her current office, she was a team leader in the Office of Bioequivalence. So following the presentation, we have a panel discussion like the other 2 sessions, and we will have additional members in the panel discussion. Dr. Andrea, Dr. Bhagwant Rege, Dr. Bing Lee, and Dr. Lucy Fang will join the speakers in the panel discussion. Subject matter expert Dr. Elisa Nikam will help me to screen the questions from the virtual attendee, and please feel free to post your question while you listen to our speakers, and we'll try to accommodate as many questions as possible for the panel discussion. And I will introduce our additional panel members before the panel discussion. So let's get started. So I like to invite Dr. Chris Bode for the first presentation.
Hello, I wanted to thank the CRCG for putting on this workshop today, and thanks to Alam for inviting me to speak. I'm going to talk about some work that we've done on in vitro drug permeation. This work was funded by an FDA contract, so I just wanted to make it clear that anything that I say is my opinion and should not be construed to represent FDA views or policies. So, as we've heard all day today, one approach to reducing the impact of NDSRIs is to reformulate drugs with an antioxidant. And in these studies, we've used a novel in vitro test system called IDOS for in vitro dissolution absorption system. And we had used this system previously in a previous FDA contract where we looked at the effect of excipients on the permeation of BCS class 3 drugs. In that project, there were no antioxidants. It was excipients in representing every major functional class of excipients, but no antioxidants. And one of the things that we learned was that CACO2 cell monolayers in the IDOS are less sensitive to excipients than in conventional transwell culture. In transwells, CACO2 cells are known to be overly sensitive to excipients. And so in that sense, they're more physiological in the IDOS, probably still more sensitive than in vivo intestinal cells. So it's still a conservative model, but more physiological than in transwells. So the objective was to use the IDOS to evaluate the impact of antioxidants on drug permeation. So this is the IDOS. It was developed and patented by Absorption Systems, which was acquired by Pharmaron at the end of 2020. So it consists of a 500 ml dissolution vessel with two 8 ml permeation chambers. And at the interface between the two is a polarized monolayer of human intestinal CACO2 cells. So the apical side of the cells faces the dissolution chamber, which has a biorelevant buffer. In this case, it's fasted state simulated intestinal fluid, or FASIF. The basolateral side of the cells faces a pH 7.4 buffer with serum albumin to model the blood plasma compartment. And this system was designed to be able to monitor both dissolution and permeation simultaneously with finished drug products. So we measured dissolution by sampling from the dissolution chamber, filtering the sample to remove undissolved API, and then measuring the concentration over time. And then we monitor permeation by monitoring the appearance of the API in the permeation chambers. So first we validated an LC-MS method for 4 BCS class 3 model drugs, and the tests that were part of the validation are shown on the right. The model drugs were acyclovir, atenolol, cimetidine, and ranitidine. And atenolol served a dual purpose. So it was a cell monolayer integrity marker, just because we have a long history of using it for that purpose, but it was also one of the 4 model drugs. So we pre-dissolved the model drugs, dosed them individually, with the exception of atenolol. So atenolol was always co-dosed with one of the other 3 model drugs. And in this study, we're only looking at, everything was pre-dissolved, so we're not looking at dissolution, we're only looking at the effect of antioxidants on drug permeation. The model drug concentrations were 10% of a clinical dose in 250 mL. Actually, atenolol was a little bit less than that. And these were the antioxidants that we tested. And I just wanted to say that this day I've learned a lot. When we first started working on this contract, the FDA group that we were working with, they said, okay, these are the antioxidants that we want you to test. And I didn't really know why, but I've gotten a sense for why that is now. It's all put into to context. So the antioxidants were alpha tocopherol, ascorbic acid, cysteine, and propylgallate. They were each tested at 3 concentrations. And I wanted to explain the rationale for the high concentration that was used. So it's been reported, and these are two of the publications, but we've seen other work presented today, that a small amount, 1% to 2% of an antioxidant in a drug product could potentially inhibit the formation of NDSRIs. So the high test concentration is based on 500 milligram dose unit weight, not a dose, but the unit weight of a capsule or tablet. 2% of that is 10 milligrams. 10 milligrams in 250 mLs is 0.04 mg per mil. So then the mid concentration was half of that, the low was half of the mid. And the amount of antioxidants was within the maximum daily exposure in the FDA inactive ingredients database. So this is the study design. On each day, we tested one model drug with one antioxidant. So the table on the right shows how this was set up. So we had a negative control with no antioxidants, the 3 test concentration of the antioxidant for that day. And then each was run with 6 replicates. So these are 6 permeation replicates. So if you remember, there were 2 permeation chambers in each dissolution chamber. So it's 3 dissolution chambers, 6 permeation chambers for each treatment. The donor medium was FASIF, pH 6.5. The receiver buffer is Hanks buffer, pH 7.4 with BSA. We collected receiver samples from 20 to 120 minutes, and then calculated the apparent permeability coefficient, or PAP, over that duration. So the drugs were dosed, remember the drugs were dosed individually, with the exception of atenolol was co-dosed with all of the other drugs. So there's actually 3 times the number of replicates for atenolol because it was present every time one of the other model drugs was run. I just wanted to give you a sense for the consistency of the model. So this is a compilation of all of the negative control data, so no antioxidants. These were run in parallel with each of the antioxidant treatments. So each colored bar is a model drug, and then the error bars are the standard deviations in all of the experiments that were done over a period of about 6 months. So it was pretty consistent. So now I'm going to show the data for the antioxidants and the model drugs, and spoiler alert, there were really no obvious effects. There'll always be a model drug on the left, atenolol co-dosed with that model drug on the right, and then the calculated PAP values in the table on the far right. There was one treatment that looked like one concentration of the antioxidant might have had an effect, but I don't think it's real, and I'll explain why when we get to that. So this is acyclovir with alpha tocopherol. So if you look at the mean PAP values and the standard deviations, it's pretty clear that there are no differences among the treatments in this experiment. So this is the one case where it looks like the high concentration might be lower than the others. So this is acyclovir with ascorbic acid, but I don't believe that one line, the light blue line, which is 0.04 mg per mil, I don't believe that's actually different. And the reason I say that is that, remember, we've run atenolol with acyclovir, also ranitidine and cimetidine. There's no sense that ascorbic acid affected atenolol in those other experiments with those other model drugs. So I think this is just kind of a random variation. I can't explain why it looks lower, but I believe that if we ran it again, probably we wouldn't see an effect on atenolol, and that tracked pretty well with acyclovir. So I'm just going to run through these slowly, and you can see the time courses and the numerical values. So this is acyclovir with cysteine. This is acyclovir with propyl gallate. Ranitidine with alpha tocopherol. Ranitidine with ascorbic acid. I wish they all looked that close. Ranitidine with cysteine. And ranitidine with propyl gallate. Cimetidine with alpha tocopherol. Cimetidine with ascorbic acid. Cimetidine with cysteine. And cimetidine with propyl gallate. So for all of the antioxidants and all of the model drugs, there was really no obvious effect on permeation. You could probably find statistical differences. We try not to do that with this type of data because the tighter the data, the more statistical differences you're going to find. But it's just kind of a quirk of the data. It may not be really physiologically meaningful. And we'll find out more as modeling is done by our FDA colleagues to determine, what magnitude of a change will translate into a meaningful difference in vivo. So for all of the antioxidants tested at the concentrations we tested, there was little or no effect on the permeation of these 4 class 3 model drugs. Suggests that reformatting drug products to include an antioxidant may be a feasible approach for reducing the formation of NDSRIs. And the next steps will be modeling, and that's all. I know that's already ongoing, to translate the in vitro data and into in vivo predictions. So this is these are my Pharmaron colleagues. This is the FDA team that we work closely with, most of whom are here today. And thank you very much for your time.
Good afternoon, everyone. And I really like to thank Alan and Andrea Raw for inviting me to this talk today, and also for the CRCG for organizing this wonderful talk. And it's really my first time to be here, and it's really honored to be selected to present this project. So today I'm going to talk about the effects of antioxidants that everyone talk about today. We want to investigate how this effect of antioxidant is going to affect the drug transporters, which are very important in our intestine. So this is a collaboration between our UCSF members as well as the FDA folks, and this is funded by the CERC project. So why are we looking at the transporter? So the transporter that I'm showing here in these figures, they are called the P-glycoprotein, PGP. Maybe many of you have heard about it. And then the BCRP, which stands for breast cancer resistant protein. These are very critical drug transporters sitting in your intestine to efflux out drugs. It is very important to efflux so that you can protect yourself from receiving cytotoxic drug. However, if something going to inhibit this transporter, you are going to have an increased drug level. So that is why it is very critical for during drug development, we need to investigate whether new drugs or even antioxidant in our situation, whether that's going to inhibit this transporter so that we are not going to affect the drug bioavailability. Similarly, for OATP2B1, it is a very important influx transporter, and it transports drugs such as rosuvastatin. If something is going to inhibit this transporter, you are not going to get enough drug into your body. So that is why it is very critical for this collaboration between FDA and our people in UCSF to investigate whether antioxidant added into a drug product, whether it's going to inhibit the drug transporters. And I listed some of the examples of this drug because they are the one that you all mentioned just now by Chris Boyd as well as other speakers. These are the nitrosamine contamination drugs. And these are the drugs that are being pumped out from by the PGP or the BCRP, as well as influx by the OATP2B1. For example, valsartan and losartan that you have heard about from other speakers today. And our question is to ask whether antioxidants used in the drug product, do they inhibit these drug transporters? Our approach is relatively simple. So first of all, we got to select the list of antioxidants. As we heard from the speakers this morning, Marco, you all selected the antioxidant from the FDA inactive ingredient. So there were 22 of them in our list, and we selected 8 others of them. They are just antioxidants found as natural products. So like ergothioneine, some CQ10 coenzyme. So those are the ones we just added to our library to make it to 30 antioxidants. And then we purchase them, and then we plate them into a 96 well plate. And we screen at a relatively high concentration, 200 micromolar. The reason why we screen at high concentration, just to make sure that if it's really inhibit, we can do further IC50. But if it really doesn't inhibit, we probably don't have to worry too much about it. It's because unlikely it's going to achieve up to that concentration. To screen this, we use radioactive compound together with the system where we can overexpress the transporter. For PGP and BCRP, they are efflux transporter. It will be difficult to screen a compound to inhibit this efflux transporter. Hence, we have to use a different system called the membrane vesicle so that we can flip around the transporter so that it's inside out so that we actually measure the uptake rather than the efflux. And we use the typical substrate that we normally use, which is the N-methylquinidine for PGP and CCK for BCRP. These are the typical substrate for the transporter. And then for OATP2B1, we just use a cell lines that's overexpressed with the transporter. And we use the substrate called estrone sulfate. And then what we do is, after inhibit it with the 200 micromolar of the antioxidant, we put it into the microbeta because these are all radioactive compound, and then we just measure how much of the substrates get into the cells. So our result here show that a list of antioxidant that is maybe very small in the screen. What I wanted to highlight is at the bottom, there are some set of known inhibitor so that we know for sure it's going to inhibit the transporter and we know our control works very well. And then what we found was these 3 antioxidants that I'm highlighting here called ascorbyl palmitate and the butylated hydroxyanisole, AP and BHA, I'll call them. Carnoxic acid is one that we added into the cells, but actually it's not in the inactive ingredient. It's one of the some other products that also act as an antioxidant, but it's not being officially listed in the inactive ingredient. So for this, because we found 2 very important antioxidants which are listed in the FDA inactive ingredient, so we move forward with further determining the IC50 of ascorbyl palmitate and the butylated hydroxyanisole. So to determine whether these antioxidants, how potently they inhibit the transporter, we perform this inhibition potency curve, where the x-axis is the increasing concentration of that respective antioxidant, for example, AP or the BHA, for the respective transporter, and then the uptake, the y-axis would be the percent inhibition. So as you expect, if you put a low concentration of the antioxidant, the uptake will be at 100% because it's not going to inhibit the transporter. When you increase the concentration of the antioxidant, as shown in the x-axis, you can see the percentage uptake decrease. So we just calculate at 50% uptake, what is the IC50? And I listed on top of that, the AP and the BHA. The IC50 is relatively from medium to high micromolar. They are not super potent, but that could be important. So what we want to ask the next question is, how do we interpret this data? It's just the IC50 value. What does it tell us? So in order to do that, we follow the FDA guidance for in vitro drug interaction studies. So this is typically done when a drug developer developing drugs, for example, investigation drug that a company do. So if they want to ask the question whether the drug that they develop in the pharmaceutical industry, whether they inhibit any of these intestinal transporter, they will first calculate the concentration of that new drug that they develop. What is the concentration that you can achieve? Divide by the IC50 that you calculated from your experiment. If that ratio between intestinal gut over IC50 is greater or equal to 10, this may suggest that the compound that you develop could be potentially inhibiting this intestinal transporter. When I say I-gut, it means I will say that the dose of the inhibitor divided by the 250 ml of the intestinal fluid that we assume. And for this case, we are doing antioxidant, so we can use the exactly similar calculation. So our FDA folks help us to calculate the intestinal concentration of the AP and the BHA. It is listed as 97 micromolar for the AP and 178 micromolar for BHA. They can be at a very high concentration in the intestinal, making some assumption what is the typical FDA oral limit dose per day, as well as what is the typical weight in a 500 milligram oral dose. So for this case, we next ask the question, what is it going to look like when we use that I-gut over IC50? How does the result looks like? So you can see that the one on the top, those are the IC50 value that I show you in my previous two slides. And if we use the exact IC50 value and then use the concentration of the AP and the BHA, we just use that, for example, the 97 micromolar, divide by the 23, which come from the OATP2B1 data. And if you divide by that, that's come to 4.2 for the AP. And then for the BHA, you can see that the ratio rank from 0.9 to 2.5. So all these values are less than 10. So this suggests to us that this AP and BHA antioxidant are probably and unlikely to inhibit the OATP2B1 as well as for the other transporter. So next, we ask the question, because estrone sulfate that I show you, these are just a model substrate. Those are not drug. But valsartan is a substrate for OATP2B1. And we know valsartan has been shown to be nitrosamine contaminated. So we want to ask the question when we use valsartan as the substrate instead of estrone sulfate, what IC50 do we get? We see relatively similar result. Usually we don't expect to see much different if you use one substrate versus another substrate for the same transporter. And similarly, we did not see much different. When we use I-gut over IC50, for the AP is 6.4 and the BHA is 8.1. And again, these are still less than 10. So it's relatively safe. I don't think the antioxidant is going to affect valsartan bioavailability. So let me show you how about those other commonly antioxidant. How does that interact with other transporter? I'm just going to zoom in this result because in my previous slide, the number is very small. I'm going to show you the list of antioxidant from ascorbic acid, which we heard a lot about today. Cysteine, we heard a lot about it as well today. Vitamin E, some other that may be mentioned as well. You can see that those values are very high. They're all above 50%. It's clearly telling you that this number is telling you that is not going to affect any of the uptake of the antioxidant is not going to inhibit the transporter. That's why the percentage is high. So it's relatively safe, and we have already used 200 micromolar, which is relatively high concentration, and which is usually not going to be that high concentration for those other antioxidants that you may have added into your drug product. So in summary, I hope that I show you that from our initial screen that we revealed the IC50 for AP and BHA, which is the more potent compound that we discovered in our initial screen. When we did the inhibition potency, it comes out to be 97 micromolar for AP and 178 for BHA. They are relatively high. And they are not going to meet the criteria for IGUT over IC50, and they are all below 10. So relatively safe. However, our future study, we will still need to perform the experiment using the actual drug substrate, especially those drugs that are the substrate for this transporter, and especially the drug has an issue with the nitrosamine contamination, such as using cimetidine. And perhaps we are going to do this experiment using our typical MDCK transwell assay. And I also like the one that Chris Boyd mentioned, the IDAS. It sounds like an amazing system that maybe we could even collaborate to use it. And then the third point I'm going to make is using our information, if we determine the next step, if any of our data show that IGUT over IC50 is greater than or equal to 10, typically, if that's going to happen, we are going to do further study, maybe using PBPK modeling or use some other in vivo experiment to show whether the antioxidant and then the drug interaction study and whether that really translates into an in vivo situation. But so far, our data is so far so good. So hopefully we'll have further update in future. So I would like to thank the members in the UCSF team, Megan and Chetan, they are postdoc in the lab, and my mentor, Kathy Giacomini and Diana Krutz, who are also involved in this project. And I'd like to thank the FDA from the Office of Generic Drugs, as well as the Office of Pharmaceutical Quality, Elam, Liang, Lei, Andrea, Bhagwant, and Dongmei, who majority of you are here today. Thank you so much for your attention.
First, I would like to thank the organizing committee to provide us the opportunity to share our work here. The topic of my presentation today is PBPK-based modeling to evaluate the impact of excipients on bioequivalence of BCS class 3 drug products. Here is my disclaimer. This presentation reflects the views of the presenter and should not be construed to present FDA's views or policies. We hear today that the nitrosamine are formed by interaction between second imine and nitrosamine ion, and one approach in controlling the levels of these impurities is using antioxidant and or pH modifiers in the formulations. However, whether these antioxidant and pH modifiers would impact the bioavailability and bioequivalence for generic drug products is currently unknown. It would be helpful to use in vitro studies, for example, the permeation testing just now introduced by the previous presenters, and in silico modeling, such as the PBPK modeling approach to evaluate the impact of excipients, including antioxidant, on bioavailability and bioequivalence of generic drug products or the reformulated products. Here is the general PBPK modeling procedure. The model inputs include the in vitro data of test formulation and reference formulation, and also the clinical data sets. And we can first use the IV data to develop the disposition model, and then use the oral data to develop the absorption model. After verification and validation, sensitivity analysis can be conducted and to identify the key factor for impacting the in vivo performance. And we can use the validated PBPK model predicted in vivo PK of batches or formulations. And then virtual BE, bioequivalence simulations, can be conducted to set clinical relevant quality attributes and simulate BE trials between reference and test products. Today, I would like to introduce you two case examples for using PBPK modeling to evaluate the impact of antioxidant on bioequivalence of acyclovir. And acyclovir is an antiviral drug, and it is also a BCS class 2 drugs at low dose and BCS class 4 drugs at high dose. The available immediately established strengths are 200, 400, and 800 milligram. And the absolute bioavailability is 10 to 30% in humans. First, we developed the acyclovir PBPK model after IV administration by using compartmental modeling and estimate disposition parameters. Our established acyclovir PBPK model can capture well the PK after IV infusion of acyclovir 2.5 mg per kg and 5 mg per kg as shown in these two figures. Then we developed the acyclovir PBPK for oral IV tablets for 200 mg and 400 mg strengths. And the solution and also the apparent permeability data can be incorporated into the PBPK model and also estimated the other absorption parameters. As shown in these two figures, the model can predict the PK after oral administration of 200 and 400 mg acyclovir IR tablets. And for validation, we use another strength, 800 milligram acyclovir IR tablets, and we use the in vitro dissolution and also the in vivo PK data for the model validation. You can see here in this figure, it shows that our built model can predict reasonably well of the PK after the oral administration of 800 mg acyclovir IR tablet. So we also validated the model using other product like 200 mg, 400 mg, 800 mg from the ANDA data, and then we can just show this here, the prediction error when comparing the predicted PK parameters versus the observed PK parameters. We can see that prediction error is around 20%. So this is reasonably well for the PBPK modeling. So we also conduct a sensitivity analysis to evaluate the impact of the permeation change on the PK parameter. Impact on the PK parameters. You can see with the increased apparent permeability change, the PK parameter ratio also changed. That with the increase of the apparent permeability, there is an increase of the PK parameter ratio. This PK parameter ratio is calculated based on the PBPK model prediction on the test product with apparent permeability change compared to the reference product with original measured apparent permeability. We also conducted the virtual bioequivalence simulations for acyclovir IR tablet. And we can see on the left figure, you can see with the 10% increased apparent permeability up to 50% apparent permeability, we can see the PK parameters and the 90% confidence interval of T over R are within the 80 to 125%. And however, when we increase the apparent permeability to 60%, we can see that the PK parameter ratio between T and R, 90% confidence interval are over the 80 to 125%. In summary, for this case example, the PBPK model was established for acyclovir IR tablet, and this model was used to assess the impact of excipients on BE parameters. Note that there is a knowledge gap on the in vivo permeability change by excipients, and we are using in vitro testing system like the IDAS system to provide the relevant information. And the virtual BE simulations were used to assess the potential impact of the excipient, including antioxidant-mediated permeation change on the BE of acyclovir IR tablets. This suggests that also more than a 60% change of apparent permeability value for test product due to presence of a certain excipient may result in risk of failing BE of acyclovir 800 IR tablet under fasted condition, similar for the 400 mg IR tablet. Another case example I would like to share is using PPK modeling to evaluate the impact of excipients, including antioxidants, on the BE of ranitidine. Ranitidine is a histamine H2 receptor antagonist. It is a BCS class 3 drug product, and available immediate release tablets are 150 and 300 mg. The absolute bioavailability is around 50% in humans. Similar like the acyclovir model, we first using IV data to develop the ranitidine PBPK model, and then using the compartmental modeling. You can see here, this figure shows that the PBPK modeling can capture the PK pretty well after IV infusion of 20 mg ranitidine. And for the oral PBPK model, we estimated the absorption parameter, and also you can see here in these three figures show that our established absorption PBPK model can capture the PK well after the oral administration of 20 mg, 40 mg, and 80 mg ranitidine tablets. We also use the other available data and from the RLD, like 150 mg and ANDA data for 300 mg ranitidine IR tablets, and to validate the model using the in vitro dissolution and PK data. We can see the prediction error well around below 20% or around, which is reasonably well. So similarly, we conducted sensitivity analysis to evaluate the impact of apparent permeability change and the effective permeability change on the PK parameter ratio. We can see with the increase apparent permeability ratio on the right figure, with the increase of this apparent permeability percentage, then PK parameter ratio increase at about over 30% increase, and then the PK ratio can be over 125%. And with the decrease of the apparent permeability for 20%, it may decreasing the Cmax ratio to 80%. So we also conducted the virtual BE simulations for ranitidine IR tablet 300 strength. On the left figure, we show with the 5% increase of apparent permeability, the PK parameter ratio is within 80 to 125%. When increasing to 10% for the apparent permeability, then the PK ratio between the T and R, and for 90% confidence interval is over 80 to 125%. Similar, if we decrease 10% of apparent permeability, the PK parameter of the ratio between the T over R, the 90% confidence interval is over 80 to 125%. In summary, the PBPK model successfully developed for ranitidine IR tablet, and the single batch simulation showed that increasing the apparent permeability by more than 30% increased the ratio of Cmax above 125%, while decreasing the permeability by more than 20%, resulting in Cmax ratio decreasing below 80%, as I just mentioned earlier. And VBE trials were used to assess the potential impact excipients like antioxidant mediate the permeability changes on ranitidine BE. And there is an existing knowledge gap on in vivo permeability change by excipients. We are using in vitro testing systems to provide relevant information. In conclusion, our GDUFA-funded research projects were conducted to evaluate the impact of drug excipients on the drug absorption and BE. The knowledge gap exists between in vitro and in vivo correlations for permeability change. As mentioned earlier, the permeability identified by in vitro system may be more sensitive than the in vivo change. The PBPK modeling can be used to predict the impact of excipients on drug absorption and the BE. The PBPK modeling may be used to extrapolate risk assessment. And the in vitro studies, the permeation testing and the PBPK modeling approach can be used together to evaluate the impact of excipients on the BA and BE of new and generic drug products and support formulation development to address a nitrosamine issue. This concludes my presentation. I would thank all the people listed here, especially Dr. Sharon Thomas, Abdullah Shoaib, who did most of the modeling work. I would like to thank Alan, Liang, and Lucy for their support, and also the absorption system work support for the permeation testing, especially Dr. Chris Bode. Thank you.
Hello, everybody, and my name is Dongmei Lu. I'm from Office of Policy for Pharmaceutical Quality, and from OPQ and CEDAR FDA. And may I have next, please? So today is my presentation that is focused on the FDA guidance of for industry. The guidance title, yes, then you know that is a control of nitrosamine impurities in human drugs. Thank you. Next one. Oh, yes, I can click. Yeah, disclaimer. So this presentation reflects the view of my personal view. So it will not be construed to represent the FDA's view or policies. And in my presentation, I'm going to provide the following information. First is the background. Second is the root causes of nitrosamine. And third is FDA recommendations to API and drug product manufacturers. And the fourth one is the most recent nitrosamine drug substance related impurity that was discovered recently. And FDA also have recommendations for the mitigation strategy for NDSRI. And the last one is reporting changes to FDA. First is the background. So what are nitrosamines? And previously, there already elaborated information on the nitrosamine. So I'm not going to go into the details. And I want to go through the next one is that because nitrosamine are probable or possible human carcinogen. Possible put right here just because in the NDBA and others, such as some other small molecules, they are all defined as a probable carcinogen. And as you know, there are two types of nitrosamines. The first is the small molecule nitrosamines, such as NDMA and NDEA. So from the structure and on the right side, you can see that that is NDMA structure. So it doesn't represent or doesn't related to the API structure. The second type of nitrosamine, it is a nitrosamine drug substance related impurity. We also call it as NDSRI. In most of cases, it could be found in drug products. And from this structure, you can tell the chemical structure on the bottom, the corner, it is related to API or to the API fragment. And here is a brief history of nitrosamine issues. Nitrosamine issue has evolved really quickly. Back in 2018, that is the first discovery of NDMA and NDEA was discovered in certain products. At that time, really a lot of recalls, withdrawals, and also drug shortage showed up. And also, next one, it is the ranitidine. Also, that is also NDMA showed up in ranitidine. Later on, all products of ranitidine was withdrawn from the market. And still in the same year, metformin extended release drug product, NDMA was discovered in that product. And following still the same year, that's really the 2020 happens a lot. And FDA become aware of nitrosamine impurity in MMP and CPNP was discovered in rifampin and rifampicin. So, you can tell that these four drug products, pretty much that is the nitrosamine was discovered in there. That is a small molecule nitrosamines. And so at that time, based on our knowledge at that time on the small molecules, FDA established the guidance on nitrosamine. That is back in September 2020. And right after that, in 2021 and 2022, NDSRI was discovered in some certain drug products, such as vanoracline and sitagliptin. And back in 2021, November, based on the information available to FDA at that time, FDA have a public announcement and try to give this recommendations on the NDSRI mitigation strategy. FDA make really greatest efforts and try to disseminate the information to industry. And FDA has this public web page for announcements, and also have a product-specific communications with industry, and such as have this IR letters or the CR letters, and also have industry meetings and to communicate and have the discussion on the potential mitigation plans, and also have workshop like this and to disseminate the information and also FDA current thinking on nitrosamine issue. Internally, there are various of working group in FDA and try to have this scientific investigation and also policy and the policy development related to this. FDA also really actively collaborated with the international regulators in these perspectives. In terms of the guidance, the first guidance was published in September 2020. The guidance was published, it was for immediate implementation because the importance of the providing timely information to API and the drug product manufacturers. And later on, in 2021, in February, the guidance was revised to try to update the information about this implementation dates. And here I try to give the contents of the guidance. And the information I present here is that where we focus on the small molecule nitrosamine. That is the content in the current version of the guidance. That includes the root causes for APIs and also in drug products other than the API sourced nitrosamines. And in the recommendations, there include acceptable intake limits, recommendations to API manufacturers and the drug products manufacturers, and reporting changes. That's including the timelines for three-step mitigation plans. And also the recommendation that's a little bit different really depends on the status of the application. First is the root causes. And for the API, the most typical theme is that it is nitrosating reaction in the route of synthesis of the API. And the second big cause is that if there's a lack of a process optimization and control of reaction conditions, such as temperature or duration of the reactions, and that is may introduce nitrosamines. The third is a cross-contamination, especially if there's a contamination between different manufacturer lines, or and if there is a third-party facility involved, such as for the solvent recovery, and that may introduce nitrosamine to the product. And the last one is the processing and the potable water. And it was noticed that in the water, potable water, and there are really small amounts of nitrosamine. So if in your risk assessment, if you don't see any risk, and you couldn't identify any, then last one, you can go to take a look at the water, whether that could be a possible root cause for your nitrosamine show up in the drug product. Root causes for the drug products. In general, the nitrite impurity in excipient that could form nitrosamine together with the at-risk APIs. And in general, it's going to show up in the manufacture process and also during storage conditions. And if there's a precursor impurity which can be carried over from API into the drug product manufacturers, and the later stage, it is going to show up this nitrosamine. And the last one is the processing and the potable water, the same reason as before. Recommendations. And the first one I want to give this information is acceptable intake limit, the AI limit. The AI limit is the daily exposure to a compound that approximates over 100,000 cancer risk after 70 years of exposure. And in this table, you can see that, and there are some AI limits for some certain nitrosamines in drug products. And we have different numbers right here. You can see there's 96, there are 26.5. In general, the smaller the number, that means the nitrosamine is more potent. And of course, this AI limit in the list in the table, that's in the units of nanogram per day. But you can also convert it into a ppm unit by the division by the maximum daily dose. FDA recommends 3 steps of mitigation strategies for nitrosamine issues. So the first one is the risk assessment. And it just show up. All right. Okay. And the first one is the risk assessment. For the API manufacturer and the drug product manufacturer, so you need to just take a look whether there's any risk in, okay, it's showing up, just to stay there. Okay. And just to see whether during any kind of a manufacture, okay, it just automatically advanced by itself though. Yeah, so in terms of the risk assessment, manufacturers need to take a look during the API and the drug products manufacturer, there's any potential risk could be identified, and even think about this degradation pathway. If there's no risk is identified, then no further action is needed. But if there is any risk is identified, then you need to go to the second step. That is the confirmatory testing. And this confirmatory testing need to be conducted as soon as possible if there's any risk is showing up. And especially you need to do that for the high risk drug product. And the third step is the reporting changes. If the nitrosamine level, it is more than the limit of quantitation, and the manufacturers need to have a control strategy and also set up specifications, in their drug products. You need to think about, the control strategies such as in terms of the manufacture process, or you want to do the reformulation, and all of the changes and set up specifications, you need to report to FDA. And I want to point out is that for the risk assessment report, and manufacturers should retain this report at the manufacturer site. You don't need to submit to FDA. And but it should be available upon FDA's request. Recommendations to API manufacturers. And these recommendations are corresponding to the root causes of nitrosamine show up in APIs. And the first one is manufacturers need to optimize design of a process in route of synthesis, should pay attention to the condition, and try to avoid amine, base, or solvent if possible. Sometimes it's maybe impossible, right? But try to keep that in mind. And also try to control reaction sequence and the process conditions. The second is that API manufacturers need to audit or monitor supply chain of the raw material, such as the starting material, intermediate, or catalyst. And the third one, try to avoid a cross-contamination, especially there is a third-party contract facility involved. And the last one, API manufacturers, of course, you can do reprocess and rework on the batches, whichever you already notice there are some kind of contamination. So if nitrosamines are detected in the API lots, what should API manufacturers do? There's two scenarios. The first one is if the nitrosamine level is more than the LOQ, the limit of quantitation, but no more than the AI limit, then the manufacturer need to develop a control strategy and including set up the specification for the API lot. The second scenario is if the nitrosamine level is more than the AI limit, and these batches should not be released unless with the FDA's agreement to try to prevent or mitigate drug shortages. Next is the recommendation to drug products manufacturers. And these recommendations are also corresponding to the root causes I mentioned earlier in the drug product. And the first one is the drug products manufacturers need to collaborate with the API manufacturers closely and just try to see whether, API, there's any possible reason nitrosamine could show up in API. So drug products manufacturers should continuously test API lot until it is verified that the API doesn't have an unacceptable level of nitrosamines. And the drug products manufacturer should evaluate all possible pathways, including the degradation during the manufacture process or the storage conditions. And the last one is try to remove the precursor impurity, such as the DMA, dimethylamine, that may be carried over from API synthesis and into the drug products, because that potentially can form nitrosamine in drug products. If nitrosamines are detected in the drug products, what should drug product manufacturers do? There are also two scenarios. The first one is if nitrosamine level is more than LOQ, limit of quantitation, but no more than the AI limit, and the drug product manufacturers need to develop the control strategy and set up specifications for the drug product. It is very necessary if a risk is inherent from API or drug products. The second scenario is if the nitrosamine level is more than the AI limit, then the batches shouldn't be released. And the manufacturer should contact FDA regarding the batches that is already distributed on the market. FDA may exercise regulatory discretion when warranted to prevent or mitigate drug shortages. And here I want to give some information about this NDSRIs, nitrosamine drug substance related impurity. And it has been discovered, identified in some certain drug products, such as varenicline and sitagliptin. And previously, we already mentioned that it's very challenging to determine the AI for each NDSRI, because that is really unique to each API. And in terms of the data, such as the mutagenicity or carcinogenicity data, that is really limited. And so in terms of the root cause of NDSRI, basically APIs which has secondary amine or tertiary amine, when they are exposed to nitrocycline compounds such as nitrite impurities in excipient, then they can be formed during the drug product manufacture or storage conditions. And back in November 2021, based on the information available to FDA at that time, FDA has a public announcement and to give this mitigation strategy to industry. Basically, 3 recommendations, strategies was given to industry. The first one is screen excipient for nitrite impurities. And the second one is add antioxidants in drug products. So that means, kind of the reformulation. And because there are some literatures already reported that a small amounts of antioxidants, when added into drug formulation, basically it can inhibit a lot of this NDSRI formation. And the third strategy is add some pH modifier in the drug products formulation, because from the reaction scheme of nitrosamine in general, it will happen under the acidic conditions. So if you add some base, as some pH modifier, that is going to change the microenvironment in the drug product. So basically, and that could inhibit NDSR formation. Of course, FDA are really open to other strategies and approaches and encourage industry to have innovative ideas and just to come and to talk with FDA, have further discussion. Reporting changes to FDA. And we have a recommended timeline for this 3-step mitigation strategy. As I have mentioned earlier, that is risk assessment, confirmatory testing, and submission of the changes. But in terms of the recommendation, there are a little bit difference. Really depends on the status of this application. The first one is for approved and marketed drug products. For the risk assessment, and that should be completed within 7 months from guidance as a first publication date. So that means actually back in 2021, March 1st, this risk assessment should already be done. And confirmatory testing, that should be conducted as soon as possible if there's a risk is identified, and especially for the high-risk drug product. In terms of the submission of changes, and along with the confirmatory testing, these last 2 steps, and that should be concluded within 3 years of the guidance publication. So that means that this year, October 1st, that is the date for this last 2-step conclusion. But because recently, and we know that about this NDSRI and showing up in several drug products, and we recognize probably we should have some kind of extension of this recommended timeline. So right now, FDA is seeking comments on this in this perspective. And if you pay attention, that was in May the 2nd, I think FDA published one federal notice and talk about this AI determination. And one question just about this extension timeline, and try to seek the comments from industry. And okay, next is for the pending applications. Pending application, there's 2 scenarios. First one is for pre-submission, and the manufacturers need to conduct a risk assessment on the confirmatory testing if needed. Changes may be submitted in amendment if it's not included in the original submission. And the second scenario is if the application is pending with FDA, then the risk assessment and the confirmatory testing is still needed. And manufacturers need to report to FDA if nitrosamine level is more than the AI limit. And if nitrosamine level, it is more than the LOQ limit of quantitation, but no more than the AI limit, the amendment, with the control strategy and the specification, that should be included in the amendment and submitted to FDA. I think that's all I have, and thank you so much, everyone. That is a long day, that is a whole day attention. Thanks. I give it back to Alan.
I'd like to give a brief introduction to our additional panel members. Dr. Andrew is serving as an Associate Director for Science and Communication in the Office of Lifecycle Drug Products within OPQ FDA. Dr. Raw is currently involved in the risk and quality informatics initiatives and is a principal architect for knowledge aided assessment and structured application, which is also known as CASA. More recently, Dr. Raw has been active in the area of nitrosamine impurities. Dr. Bhagwant Rege is the Division Director for the Division of Biopharmaceutics in the Office of New Drug Products within OPQ FDA. His division at FDA is responsible for assessment of clinically relevant in vitro release specifications for drug products, in vitro in vivo correlations, physiologically based biopharmaceutics model, scientific bridging strategies, biowaivers, and BCS classification request. Dr. Bingley is serving as an Associate Director for Science in the Office of Bioequivalence, OGD FDA. In her role, Dr. Lee provides scientific leadership and expertise for the assessment of bioequivalence studies and oversees the scientific program, including guidance development and implementation in the Office of Bioequivalence. Dr. Lucy Fang is serving as the Deputy Director for the Division of Quantitative Methods and Modeling, that is also known as DQMM, in OGD's Office of Research and Standards. Dr. Fang has established herself as the FDA expert in the use of quantitative clinical pharmacology approaches in the review and regulation of generic drug products. So we have received a couple of interesting questions. First question is, in general, reformulation by adding new excipients in the solid oral dosage form would be categorized as level 3 change in super guidance, for which FDA usually require a full bioequivalence study. Does FDA open for applying alternative approach such as biowaiver for nitrosamine impacted applications?
Maybe I can get started. I feel that we have gained some scientific basis to think about alternative approach for the nitrosamine impacted applications. Actually, I think 3 considerations should be put together if we think about alternative approach. One is the research and lab result that we gained since the nitrosamine issue was, evolved in 2018. From today's discussion, we heard that we've explored variety of different root causes for the nitrosamine emerging, and we also gained some, understanding on various approaches to mitigate the nitrosamine formation. We also have validated analytical methods to quantify the nitrosamine. And in parallel, we also, in exploring, getting more understanding of the AI of the intake of nitrosamine. So this would serve as one of the primary sources that we can think about as a base for alternative approach. The second consideration would be our fundamental understandings and basic knowledge of the drug product. Okay, physical chemical property of the drug substance, for example, its permeability, its solubility, as well as the mechanism of drug release, whether drug product is IR product or MR product, what is the mechanism of drug releasing, and etc., right? And the third knowledge that we should put together about alternative approach would be the existing regulatory pathways that we currently have in our FDA's practices. Okay, for example, we have SUPAC in place, right? SUPAC has recommended, for a certain level of change, what bioequivalence package that you should put together. And also we have BCS guidances and etc. So putting all of these knowledge together, I think we have a science base to consider a more flexible and also adaptable alternative approach. And I also want to encourage the industry to work together with industry, with FDA, and also, collaboratively develop alternative approach for this nitrosamine impacted applications. Thank you.
Thank you, Dr. Lee. Anyone want to address that question? Okay, go ahead.
Yeah, I really want to echo what Bin just said. Like FDA is opening to working with our industry colleagues to identify some opening some door for some alternative approaches with adequate justifications. So today, in the opening remarks, I think Dr. Robert Leinberger nicely elaborated that modeling simulation can play a significant role as part of the justification for alternative approaches. So today, we really heard different kinds of modeling. For example, in the morning, we heard the kinetics model to predict the risk of nitrosamine formation. Then we heard the QSAR model, and then we heard the physiological-based pharmacokinetic model. So, those, from my personal view, I think those models, that if we really incorporate some mechanistic understanding into this modeling, can really provide some very important, the important information and basis as part of the justification. Of course, the important thing is we have to have solid data to really validate the model's predictive ability. So that's really come down to the essence of the justification. So thank you.
Thank you. I have two questions that is related to policy or guidance, maybe. So FDA has updated nitrosamine guidance recently to specify timeframes for completion of 3-step mitigation activities. That is mentioned in Dr. Dongmei's presentation, that is risk assessment, confirmatory testing, and reporting changes. Does FDA have any plan to extend them? And the relevant another question is, does FDA have any plan to update the nitrosamine guidance to include NDSRI information?
So first one is in terms of the timeline for a 3-step strategy. Because at the time, when we have this recommendation in terms of the timeline, that was just based on the information of a small molecule nitrosamines. So that is the reason why we established a timeline by that time. But as you already know, nitrosamine issue evolves so quickly. And now we already have this NDSRI, and it is going to have a really much bigger impact than the small molecule nitrosamine. So, so in terms of the timeline, we definitely consider that, because we talk about this different strategies for the mitigation, right? And especially for the reformulation, and that is going to take a lot longer time. And also, if you consider bioequivalence or bioavailability testing, and that is going to take additional time. So definitely FDA and puts all the factors together, and we really have this consideration about this timeline, but for how long it is going to be extended, as I have mentioned in my presentation, and we already have one question and to the public comments, and that is in the May the 2nd, the Federal Register notice, that is related to the AI determination, but we do have one question. So if you have any thoughts or suggestions, you can just go there and provide your comments. So this is the first one in terms of the timeline. And the second one, in terms of this, the revision of the guidance to add NDSR information. Yes, so basically the NDSR information, FDA is really actively engaged in this, scientific investigation for different drug products, and also, of course, and work closely with industry. And second, and we also want to develop some kind of a policy or recommendation, and you will see within FDA, or also collaborate with some other universities, research institution. We have some kind of a research project going on. So all of this information, and we want to just come back together to form and formalize this, what will be the best and appropriate recommendations related to NDSR mitigation. Yes, so definitely, yes, that is in our mind priority. Yes, so no worry, and we're going to revise this guidance, and but it's going to take some time because in terms of research and in terms of this policy development, and it's going to take some time. Yeah, thank you.
Thank you. Another question is that we have seen from session 1 speakers that pH modifiers are also very effective in controlling the formation of nitrosamine. So the question is, do we know or does FDA have any in-house data or evidence or any active research that suggests that pH modifier doesn't have any potential to alter the permeability of a drug product?
I mean, if the reformulation only involves addition of small amount of pH modifier, which typically is going to be a basic compound like sodium bicarbonate or potassium bicarbonate, I mean, you can imagine after ingestion, it's just going to hit stomach acid and get neutralized. So just based on that, it's very highly unlikely that it's going to affect permeability, but that's just intuitively that I can say.
Yeah, but I would also add to that, though, however, is that if you do, add those pH modifiers, the principal risk you're going to have is just it's going to alter the pH microenvironment of the API, so that could impact the drug release and it could impact the solubility. So probably the key here is to ensure that should you use the pH modifier, that it does not impact drug in vitro release. That's the most significant, failure mode risk in terms of the bioavailability. So that's something that should certainly be investigated.
Thank you. So this question is for Dr. Chris Bode for his presentation. So in vitro permeability study also can be conducted using transwell system besides the IDA system that you have used. So what are the advantages to use the IDA system for excipients? Can you also discuss the translation of in vitro IDA's data to human subject in vivo?
Yeah, as I mentioned that in transwell culture, the conventional way of using Keiko2 cells, the cells are often overly sensitive to excipients, and that's because the excipients can settle on the surface of the cells, and with IDOS they're oriented vertically, so that's not possible. And there's also more dynamic mixing. It seems like in IDOS there's still more sensitive to excipients than in vivo. So it's still a conservative model, but definitely more physiological. And I'm sorry, what was the second?
So how to translate the in vitro IDAS data to human subject in vivo?
Yeah, that's something. So from the first FDA contract that we worked on with other excipients, Fang's group has been doing PBPK modeling, and I think that's what will be required for this data also, to see, like, what if there was a change, what magnitude of change would translate into a difference in absorption in vivo.
Thank you, Dr. Bode. So this question is for Dr. Fang. So you have shown the use of PBPK modeling to assess the risk assessment for the inequivalence for the reformulated drug product. So what are the recommendation or consideration we should take on incorporating the in vitro testing result, like permeability or the IC50 values for the transporter to develop the model and in predicting the in vivo performance of non-Q1, Q2 BCS class drug?
Yeah, so first, I think we need to know what is the mechanism of absorption of this drug product and what is the rate limiting step. If it is a permeability, and we also need to understand whether this is a passive permeation or active permeation through transporters. Then the relevant in vitro testing need to be conducted. After that, that in vitro testing data can be incorporated into the PBPK modeling and to evaluate the impact of this excipient on the in vivo performance or the BE. So that is the consideration for the validation of the PBPK modeling. Ideally, we need to have the in vitro and the in vivo data set to be put into the PBPK model and see whether that can capture the PK, as I mentioned in my presentation. And then also another note is that the degree of the impact of the excipients on the permeation and then on the absorption of this drug product can be drug dependent. The sensitivity could be different depending on the drug property. Yeah, that's all from me.
Thank you, Dr. Fang. So another question is that how can you please explain how to choose the concentration of antioxidant for the permeability and transporter assay or other in vitro risk assessment assay? Maybe this question maybe can answer by Dr. Sook or Dr. Chris.
So for the in vitro assay, when we select the concentration of the antioxidant to screen, we have to balance between the solubility of the antioxidant so that it makes sense that we could screen that concentration. I know in general, we don't really screen more than 1 millimolar. That's definitely an issue. So we typically go between 100 to 500 micromolar is relatively typical for us in terms of screening inhibitor for transporter. So that's the range that we normally use. We were trying to be more careful in the sense that we try to go as high as possible. And because in the intestine, we know that the drug can be given at very high doses, some of them, but not all of them. But we just want to maximize it so that we don't over limit the solubility. So 200 micromolar to us is in general is typically a concentration we will begin with, because we know even at that concentration, maybe it does not make sense to be that potent. But then because of the initial first high throughput screening, when we select the cutoff value, anything that's inhibit more than 50%, we still do a separate experiment to get the whole complete IC50 curve. So we know that we already try to avoid getting false positive or false negative result by selecting that. Based on our knowledge for transporter assay that we have screened over several years in Professor Cathy Giacomini lab. So that's how we select.
Thank you. I like to see if okay, go ahead, please.
Sorry about having so many questions. One of the things, Andre, if you remember last year when we were talking about quenching of the nitrites, etc., a topic that comes up from the regulatory perspective, since this is dominated by the generic, is the pharmaceutical equivalence versus pharmaceutical alternative. When you have a hydrochloride salt and you have added ascorbate or something else, you might bring into question the pharmaceutical alternative. Are you making a pharmaceutical alternative in that process? If you adjust the pH, a hydrochloride could become free base. So those are regulatory issues that need to be, I think also thought, even though I'm an ardent believer in, blocking nitrosation, but those are also thoughts that should come. One question that I was having on the, when we were talking about the addition of the antioxidants, and Bingley, you mentioned SUPAC. The question is, of course, SUPAC doesn't include anything equivalent to an antioxidant. So it would be great if, SUPAC is also old, to revisit it and maybe add addendums like, or like the ICH, R1, R2, R3. It would be a great idea if that comes with the data. The other thing I was going to talk about is in the studies that you did. I was very surprised with the choice of valsartan, because valsartan is one of the few drugs where the nitrosamine is not coming from the drug. It's a solvent with a reagent, sort of. So adding of an antioxidant really doesn't do too much to valsartan. So I was a little surprised about the choice. Was there any reason you chose it, or just because it was a popular first-in-class situation?
Actually, I did not know that. I'm glad you bring it up. I used valsartan because it's in FDA
As
website.
I said, it was the neon light one.
yes.
Valsartan is one of the few drugs where it's a solvent that interacted with nitrite that's used. So, a nitrite inhibitor would never help that. In the same token with ranitidine also is an out-of-the-world standalone drug where, there is an intermolecular reaction going on there. So probably adding of, it is probably one of the strangest choices, yeah.
Yeah, so these drugs were not necessarily chosen for their ability to form NDSI, but they were model drugs that represented that came under BCS class 3 or substrate for specific transporters that we were trying to investigate on regarding effect of antioxidants. So they were just model drugs.
Models,
Yeah,
I understand. And then the last topic I wanted to say is we are so heavily oriented towards oral drugs, and that to IR. When there is a capsule with a multi-coated bead, trying to inhibit with something is very difficult. And so that is a big thing. And also let us not forget injections, where adding of something will make it non-Q1, Q2, and hence not acceptable. And topical drugs, we don't know how these NDSRIs or even the small ones behave. There are quite a few topical drugs which may involve, not as many as orals. But I think I just wanted to bring this to the notice that this should also be looked at.
I think that's a good point. There are a lot of possible scenarios where the current regulatory pathway is not covering, and those missing pictures are the risk part that we're taking. Okay, so we mentioned, we have some science, we collect the data, we have existing knowledge, scientific principles that would only weigh on the science part, but there is going to be a risk part that we are facing too in addressing this vast number of nitrosamine issue that is pressing to us that need to be addressed as soon as possible, inevitably. Yeah, thank you.
I see another question.
Yeah. Can you, oh yeah.
Yeah,
it's working.
we can hear you.
Thanks today for the very interesting presentations that you provided. I found them fascinating. I have two, I guess, policy-related questions, specifically with respect to the guidance. I guess the question, I'll ask the question once and then explain the two. Whether there's been discussion or consideration about making changes to these two items, and now I'll explain them. The first one is the requirement that any validated analytical method for a nitrosamine have a QL of not more than 30 parts per billion, 0.03 ppm. If memory serves me correct, sometime around February of 2019, FDA issued to any ANDA holder for a sartan a guidance about how to develop the analytical methods. It made sense at the time because of maximum daily doses for the drugs, and because there some struggle amongst laboratories to achieve QLs necessary to be appropriate for the specification. And then, when the guidance was issued, 8 months or so later, that was entrenched for all products, all nitrosamines all the time. It certainly makes sense for high maximum daily dose drugs like metformin. In fact, in some cases, it needs to be lower than 30 ppb. But it creates, I think, a bit of an analytical challenge doesn't necessarily have to be there for very low maximum daily dose drugs. For example, if it's less than 1 milligram, you can end up having, even for something like NDMA, a specification that's tens and tens of ppm, but then you need to have a validated method spanning all the way down to 30 ppb. So just food for thought about whether that's something that could be considered to be changed in an upcoming revision to the guidance. And then the second one I have has it's something akin to that. It's the requirements when there are multiple nitrosamines present in the drug product. Right now, the FDA guidance requires that the sum of all nitrosamines shall be less than the value allowed for the most potent one. And again, I can understand when it was being conceived why that was the case. Also, there wasn't a big, I see Andre nodding his head, yes, there wasn't a big dynamic range between high potent and low potent. It was 26.5 or 96. Makes sense. The spirit of the guidance, as I understand it, is that the nitrosamines present shall not increase the lifetime risk of the patient by more than 1 in 100,000. Of course, that's proportional to the potency of each nitrosamine. I think you're aware that the EMA and Health Canada guidances allow dual choice, either the FDA choice or just do not exceed the 1 in 100,000 lifetime risk, which will become more significant based on all the presentations today about establishing AIs for NDSRIs. I think we all agree what's going to come to pass for some of them is they're going to have pretty high AIs relative to NDMA and NDEA. In such a case, the 1 in 100,000 option would make a lot more sense. Anyway, those are my comments, and just wondered if anyone wanted to respond to them.
Okay. Thanks for the question. So the first one, in terms of the analytical method, the LQ requirement, 0.03 ppm, yes, you're right. In terms of the history and EMA and FDA at that time developed just based on certain drug product. And that totally makes sense at that time. And so right now, we already see there's different drug products with this different MDD. And really that is if the MDD, it is really small or really big, and though you can see significant difference. So we do have some kind of consideration. So if you see some kind of a scenario like this, and please provide a certain justification. And if for more than 0.03 ppm, and or you couldn't, such as you couldn't reach 0.03 ppm, and this is really adequate information of MDD, and how do you see, and that is going to be viable for your drug products with this as analytical method sensitivity, and provide this information to FDA. Yeah. That should be fine. And the second part is talk about this total nitrosamine limit, right? And yes, the current guidance, we talk about if there's more than 2 nitrosamines showing up in one drug product, then the most potent one, that should be the limit for this total limit. And we realize that there are some cases, especially and such as this 2 or 3 nitrosamines with the AI limit really have a significant difference in terms of the magnitude, such as one, it is maybe 1,000, one 1,500, and the other one is 26.5. And sometimes may not be practical to, just make every nitrosamine and go back to this, to the most potent one limit. That might be impossible sometimes, if you do have this kind of a situation, and just you can come to talk with FDA. Yeah.
Thank you.
So I have another question. So for the permeability study, the permeability model, it may be more relevant for the BCS class 1 and 3 drug that has low permeability but high solubility. but what approaches would be more relevant for the less soluble drug product like BCS class 4 and BCS class 2? Anyone?
I can take a crack. So BCS class 4 drug it's a difficult situation, right? Because you are limited by both permeability and dissolution. So certainly if you have any alternative approach that you wish to propose, you can come and talk to FDA. That's the extent of what I can say for that. BCS class 2 drugs, again, you have low solubility and high permeability. So it is very unlikely that antioxidants are going to affect permeability, and that could potentially, only based on permeability change, that you might have difference in exposure. So in that case, what probably one should try to make sure is you're not changing the release rate. Addition of certain excipients won't change the release rates. But again, any alternative approach, feel free to come and talk to us in your submissions or meeting requests, and we can take a look at it.
I have a question, actually.
Sure.
Does do nitrosamines, or NDSRIs, do they cross BCS class boundaries? Is it all, do you see them with all different classes, BCS classes of drugs? I mean, obviously there's some class 3, but I don't know about others.
It depends on the structure, right? So if you have that structure, moiety, secondary amine compound, that could be present in any BCS class typically. So
Thank you. We are almost actually little bit over than the allocated time. So any further question from the in-person attendees? If not, then I want to thank all our panelists for this interactive discussion. And right now I want to invite Lucy Fang for the closing remark. Thank you everyone for the wonderful attendance and presentation discussion. We really enjoyed that, right? Thank you.
Okay, I know it's 5:30 and you want to go home, so I will be fast. So as we approach, as we wrap our today's workshop, I really want to take this opportunity to thank all our speakers and panel members. I also want to thank CRCG and directors of CRCG, Jim and Anna, for hosting this hybrid workshop and providing the necessary logistics that really helped us in organizing this workshop successfully. A big thanks to Alam and Sam from OGD, Andrew Raw from OPQ, and each member of the organizing committee to arrange such a wonderful workshop that covered quite comprehensive topics throughout different sessions. Through this workshop, we are able to bring different internal stakeholders within FDA, as well as many industry and academic speakers. I'm glad to see the strong interest in this workshop. We have about 2,500 people registered for this workshop. And this also tells us how important this nitrosamine impurity issues is to the regulatory agencies and the drug manufacturers. One of the challenges for developing the effective mitigation strategies of the nitrosamine impurity formation is that we have limited data that would allow us to understand the root cause of the nitrosamine formation, assess the risk of nitrosamine to our public health, and identify the most effective effective mitigation strategies to prevent the formation of the nitrosamine. We have seen few great research articles at individual institution level, but we need more research and investment to this sensitive public health issue and make the research outcome public. We need collaborative efforts from regulatory agencies, industry, as well as the academia to handle this issue effectively. the great scientific presentations and the panel discussions that we had today will help us to understand the problem better and will stimulate the new and effective ideas to mitigate the risk of nitrosamine formation. I hope you enjoyed our workshop today and wish you a very good evening. Thank you.